In the last video, we explored how we go about isolating autoinducers, that is, the molecules that are produced by bacteria that once the bacteria, and hence the autoinducer, reaches a certain concentration, trigger coordinated group behaviors amongst the bacteria. So at the end of the pipeline of isolating an autoinducer and identifying it by NMR, mass spectrometry, and other analytical techniques, what general types of chemical structures result from this? There are a wide variety of different chemical structures, but for the most part, we can break those into 3 classes of autoinducers that are most common. We'll go through each of those 3 classes in this video. The first class is what's referred to as the acyl homoserine lactones, or AHLs for short. So AHL stands for acyl homoserine lactone. As an example of an acyl homoserine lactone, what we can draw out is a lactone molecule. Lactone, remember, is a cyclic ester. So I've drawn an example of a cyclic ester right here. That is our lactone portion of the molecule. And then we additionally will generally have an amide group branching off here. And then a carbon chain here, which I'm going to denote as R here, where R differs in the length and substitution on this N-acyl chain. So in different species, what we generally see is that the length of this R group, where it's generally primarily a carbon chain here, the length of that carbon chain varies by species, allowing individuals to recognize members of their own species from other species based on this different chain length, as well as the substituents that are on this vary as well. And when we say the substituents vary, what that means is that within that carbon chain, in many cases, there are things like carbonyl groups interspersed and groups of that sort. And ultimately, this chain here allows species to recognize members of their own species. The acyl homoserine lactones are primarily used by gram-negative bacteria. So if we look across gram-negative bacteria, such as E. coli, Vibrio species, which are producers of a lot of bioluminescence, Pseudomonas aeruginosa, which is a gram-negative bacterium that is prevalent as an infections of the lungs of cystic fibrosis patients. Primarily, what we'll see is that acyl homoserine lactones are the main type of quorum sensing molecule autoinducer that is used there. So these are primarily for gram-negative bacteria that will communicate using this. For example, as a specific molecule, the organism referred to as Vibrio fischeri is the Vibrio that exists as bacteria in the light organ of squid, and this is the bacteria that bioluminesces to make the squid emit light. And so if we take this acyl homoserine lactone and we add that to cultures of the Vibrio bacteria, what we'll see is that the Vibrio bacteria will begin emitting light regardless of the concentration of bacterial cells. Or if we use a Vibrio fischeri that has gene deletions so that that organism is not even able to make this molecule on its own, but instead we supplement that molecule into the cultures, suddenly the Vibrio fischeri will gain the ability to produce the bioluminescence. So this is an example of one of the many hundreds of different AHL quorum sensing molecules that have been described. And you can see in the chain here that the carbon chain, we could determine the length here, and then the substituents we have a carbonyl group there, or this ketone group, that distinguishes this particular autoinducer from autoinducers recognized by other species, such as recognized by E. coli, so that each individual bacteria has its own acyl homoserine lactone or lactones that it uses to recognize other members of the same species and coordinate its group behaviors. So as the second type of class of molecule that's common as an autoinducer, we will look at peptides that act as autoinducers. And these are used by many gram-positive bacteria. And we're going to record these as modified peptides, because what we generally see is that a peptide chain of some sort has been modified with one or more functional groups or cyclization in order to create a quorum sensing molecule, or otherwise known as an autoinducer. So in these modified peptides, this is typically the language used by gram-positive bacteria, analogous to how gram-negative bacteria were using acyl homoserine lactones. Gram-positive bacteria use modified peptides. And much like there were a variety of acyl homoserine lactones that act as quorum sensing molecules for individual species of gram-negative bacteria, gram-positive bacteria with their modified peptides exhibit a wide variety of different diverse structures as well. So as one example of that, we could write out the one-letter code here for a representative gram-positive bacteria modified peptide. So writing out the one-letter codes of the individual amino acids here. And if we left it just as this, as I've written here, this would just be a standard peptide chain like we've seen a few times throughout the semester, for example, when we're looking at proteins in our first unit. But in this case, there are generally additional modifications that take place. And for this particular example molecule, what happens is that the methionine residue forms a bridge via a thioester bond to the cysteine residue. So what I'm showing here is that there is a link between the cysteine and the R group of the cysteine is the sulfur, the thiol group. The thiol group reacts with the carboxylic acid group of the methionine terminus here to give this linkage to create a cyclization within this molecule that enables the recognition of this compound as a autoinducer. And we could write that out a little bit more specifically rather than just showing the one-letter codes and some atoms coming off there, and zooming in on what's happening here in the reaction between our cysteine residue and our methionine. If we have our cysteine residue, keep in mind that the cysteine has that thiol group over here. So this would be our cysteine. And keep in mind that in the case of the cysteine being part of a peptide chain, we don't actually have here a hydroxy group in our carboxylic acid, but instead that continues on in the chain of the amino acid to form a peptide bond. Likewise, at our nitrogen atom right here, rather than having NH2, if it's part of a long peptide chain, instead what is actually present there is just NH, and then that connects over to the carboxy group of the next amino acid in the chain. So the part of this molecule that is going to be focused on in the reaction that's occurring here to make this bridge connecting the two amino acids there is the sulfur atom is going to be reacting with the methionine carboxy group. So our methionine, if we draw out the chemical structure of that, remembering our amino acids here, I'm going to put just a squiggly line here to represent where the methionine connects to the rest of the protein via a peptide bond. And here at the C terminus of the protein, we would have our carboxylic acid group. And what will happen is that the sulfur atom of our cysteine acts as a nucleophile, coming in and attacking the carbonyl group here to ultimately form this link that we see between the carbonyl group and the sulfur to give this thioester connection that is essential to the biological activity of this peptide. So by modifying this peptide, the peptide becomes a quorum sensing molecule, an autoinducer that is used by gram-positive bacteria. Then our third type of autoinducer that we can focus on here is a structurally unique one that took scientists a rather long and painful time to discover because of the fact that it contains a boron atom, which is a very unusual atom to exist in a molecule produced by a living thing, because boron is a relatively rare element within the natural environment. And so most organic molecules produced by living things don't contain boron, and as a result, it took scientists quite a long time to identify this particular molecule, which is referred to as AI2, autoinducer 2. So we'll refer to this as AI2, which refers to autoinducer number 2. Autoinducer 2 is unique relative to the other types of autoinducers that we refer to, the acyl, homoserine, lactones, and the modified peptides, in that this is considered to be a so-called universal signal. It is widely distributed amongst a variety of different species of bacteria and is thought to play a role in coordinating behaviors not amongst members of a single species, but in coordinating more broad responses amongst a variety of different bacteria that are present in a particular environment. So this is referred to as a universal, and I put universal in quotes because it is certainly not the case that all bacteria recognize this quorum sensing molecule, but a wide variety of them do. And so as a result, it's been dubbed a universal signal for quorum sensing, or a universal autoinducer, because of its wide distribution across different species. And so let's go ahead and draw out the structure of autoinducer 2 to illustrate how it is unique. And I'm going to plug in that bromine first and foremost, that boron first and foremost, because boron is a rather unusual atom to see appearing in organic molecules that are produced by living things. And so drawing out the complete structure here, we have a bicyclic molecule with some hydroxy groups attached here. We have an ether group appearing here within this second ring as well. Drawing out the complete structure and finishing this up, the boron is directly connected to two hydroxy groups here and here. And if we were to calculate the formal charge on the boron, we'd find that it has a formal charge of -1. So by techniques such as carbon-13 NMR, proton NMR, and a variety of two-dimensional NMR techniques, the structure of this molecule remained unclear, in contrast to generally the structures of those other types of quorum sensing molecules, the acyl, homoserine, lactones, and the peptides, being readily determined by 1D and 2D NMR techniques. Instead, 1D and 2D NMR techniques turned up short in determining the full structure of this molecule, and the reason was that this boron atom was unexpected, and the NMR techniques were not capable of discerning that there was a boron atom here. And so ultimately, it was through crystallization that this compound's structure was identified. And in fact, interestingly enough, this particular compound was identified by crystallizing it in association with the protein receptor that binds to this protein, that binds to this autoinducer, in order to trigger the biological response. So rather than the scientists being able to crystallize just this boron-containing autoinducer by itself, instead, in order to accomplish crystallography and identify this compound, it was crystallized within the active site of the protein that it binds to. So this is my lousy attempt at drawing a protein that receives this molecule. And so it was actually a co-crystal structure, meaning a crystal structure of multiple things, of the protein receptor that this autoinducer binds to, as well as the protein. And so that study gave information about not only the structure of the protein that receives this signal, but also the structure of this chemical signal itself. So now that we have evaluated the three main classes of autoinducers, those being acyl homoserine lactones, modified peptides, and autoinducer 2, what we're going to do in the next video is look a bit more closely at how these molecules are assembled by bacteria, particularly looking at how acyl homoserine lactones and modified peptides are produced by bacteria. So stay tuned for that.